Today I want to talk with you about some of the work that I've done during my PhD in the development of computational genetic approaches for the dissection of complex traits. And I'll start with a nice, or I hope a nice, high-level introduction, which I'll motivate with a very simple question. Why do we study genetics? So I think as a computer science PhD student, it might make sense for me to be asking this from a perspective as a computer science PhD student. Why do we, as computer scientists, study genetics? And I think I'll answer that question when I talk about my individual research projects. But right now, I'd rather ask it from a different perspective, from the more general perspective. Why do we, as scientists and as human beings, as individuals, study genetics? And the first answer I'll put forth is that genetics is deeply intuitive. And I think this deep-rooted intuition that we have about it is very nicely shown in this somewhat comical picture, showing us that the traits of a child are really just a mosaic of the traits of their two parents. But secondly, and perhaps more importantly, I think that genetics is deeply personal. So this is a picture of my grandparents. It was taken in the late 1940s. My grandfather is 24 years old. My grandmother's about 20. They had just gotten married. They hadn't had any kids. They certainly hadn't thought about grandkids. And I very much doubt they had ever even heard the word genetics in their entire lives. But regardless, they still knew something about it because of their personal experience. They had seen their own traits be inherited down from their parents and from their grandparents. And they knew something about the disease risks that they inherited. And they knew about the potential to pass those on to their children. And perhaps much later, maybe in 1981, let's say, to their grandchild. And of course, now we know that the way that traits are passed from one generation to the next has something to do with this interesting molecule called DNA and a whole host of phenomena that surround it. And moreover, we know that the similarities that we observe between individuals and the differences are correlated with similarities and differences that we can observe between individuals at the DNA level. So that is, variations in DNA are sometimes directly correlated with variations that we can observe in traits. And so this very simple yet powerful idea has been utilized in the past few years to develop an entire technology known as the association study. And the basic idea behind association is this. Let's collect a number of individuals that have a disease, we'll call them the cases. Let's collect a number of individuals that don't have the disease, call them the controls. And then for a particular genetic mutation, let's look to see how many of the cases have this mutation versus how many of the controls. So in this particular case, we see that only about a third of the controls have this mutation, while 3/4 of the cases have it. So if we saw this, it would indicate that if I have this disease, I'm more likely to have this mutation. Or conversely, if I have this mutation, I'm much more likely to develop this particular disease. We can do something similar for quantitative traits. And the way that works is this. We take our individuals and we split them into groups based on their genotype. So we have individuals that have 2 copies of an A, individuals that have 2 copies of a C, and individuals that are A and C. And then we simply plot the genotype by the phenotype, and we look for a statistical correlation between the two. In this case, we see that for every copy of the C allele, I seem to have an increase in my phenotype value. So if I take this idea and I apply it across the genome, that is, if I apply it to every variation that I can observe, we start to see something like this. And we call this a genome-wide association study, or a GWAS. So what you're looking at is on the x-axis, we have the genome, chromosome 1, 2, 3, and so on. Each dot represents one genetic variation we can observe. And the height of the dot represents how significantly associated it is with a particular trait. If any dot that passes this line is considered to be significant. So if we look at this one association at a gene called ApoA2, we see it's very large, which means it's very significant, and it likely has something to do with modulating this trait. If we apply this to many phenotypes or many traits, we start to see this picture emerge. Now what you're looking at is a karyotype sort of image where we have chromosome 1, chromosome 2, chromosome 3, and so on. And a dot has been placed at every location where a significant association has been identified for a trait coming from one of these categories. And so what you see when you look at this is that there's been many, many associations identified all across the genome for a whole host of different traits. And this picture, which is really the combination of hundreds of studies, took many, many years to create, and probably cost around hundreds of millions of dollars, is a very important resource for the community because it allows us for the first time to really start to delve deeply and understand at a low level the genetic basis of complex traits and really start to be able to develop therapeutic interventions for diseases. And it also allows us to start making genetics very personal and predicting our risk for certain diseases and our children's risk for certain diseases. But regardless of all these advances and all this promise, there's still a lot missing. In particular, there's many traits for which we know there's a genetic basis. However, we haven't been able to find many significant associations. On the other hand, there's some traits for which we found many significant associations, but we're not able to accurately predict a trait for an individual given their genetics. And so to understand why we might be missing so much, I think we need to go back and think about the assumptions that we're making when we're performing association analysis. In particular, we need to realize that when we use association, we're assuming a very simplistic model for genetic systems. In particular, we're making the assumption that one genetic variation, or one SNP, has a direct effect on a trait, or maybe that something that's correlated with the SNP has a direct effect on the trait. But we know that this is an oversimplification at this point in time, and that a more realistic model for genetic systems probably looks something like this. So we have a SNP. It has an effect on a very complex network. Inside of this network, we have many phenotypes interacting in a complex way. We have other SNPs affecting those phenotypes. And this entire network is conditioned on an environment such that if we were to alter the environment slightly, we might alter large sections of the network. And any trait we're interested in is really just one node inside of this complex network. So although the SNP may affect the trait, its effect will be mediated in a complicated fashion. So the way I like to think about these two models is that they exist at the ends of an entire spectrum. So on the one end, we have this simple model. On the other end, we have this very complex model. And moving along this direction of increasing model complexity, we have many intermediate models for which we can use to evaluate the relationship between genetic and phenotypic variation. So you might say, well, if we're not getting what we want out of the simple model, then why don't we just jump to the other side and start using something more complex? And I think the answer to that is pretty simple. In order for us to do science, we need to be able to design studies. And in order to design studies, we need to be able to ask certain very relevant statistical questions. So for example, when I use this model, I know how to ask and I know how to answer the question, what sample size do I need in order to identify a SNP that has a certain effect on a trait so I can achieve a certain statistical power? On the other end, it's very difficult for me to answer this question. In other words, it's very difficult for me to figure out how many individuals I need to go out and collect so that I can discover a network of unknown complexity, and then on top of that, to find a SNP that affects that network. So one of the things I wanted to do during my PhD was to move away from this simple model in a very careful way so that I could examine the relationship between genetic and phenotypic variation in a way that was more aligned with a realistic version of genetic systems, while at the same time maintaining this very important ability to ask certain relevant statistical questions. And in doing so, enabling the next iteration of discovery. Or maybe we'll call it GWAS 2.0. Okay. So today I'm going to talk with you about three projects. Oh, sorry. Sorry, animations.
We'll edit it out.
Yeah, we'll just edit it. Okay. So today I want to talk with you about three projects that fall into this context. The first is a multiple phenotype association method, in which I introduce a mathematical and computational construct I call the matrix-variate linear mixed model. In the second, I'll introduce a meta-analysis method for model organisms. So many of you may know that model organisms typically exhibit a large amount of population structure. And this population structure can cause major problems when trying to perform basic association analysis. You can think of this problem being compounded if you were to try to combine multiple populations through meta-analysis. And we deal with that problem in this project. And in the third project, I'll introduce to you a method for calculating gene coexpression in such a way that's robust to a particular type of statistical confounding that we find in microarray experiments. Okay, so I'll move on to my first project, multiple phenotype association. And for every one of these, I tried to give kind of a high-level graphical intuition. So the graphical intuition here is that the goal is we want to identify SNPs that have an effect on multiple traits. So here I have trait 1, 2, 3. I know they're correlated in some way, and I want to use the information about the correlation structure between these traits in order to identify SNPs that affect all of them. So if I try to extend this cartoon version of the association that I showed you previously, where I'm trying to find a correlation between the genotype and the phenotype, we can extend this to multiple traits in the following way. Let's say we have phenotype 1 and phenotype 2, and we plot them against one another. And then we color each dot with respect to the genotype for the individual contributing this measurement. What we see when we do this is two things. First, we see these phenotypes are correlated. And secondly, we see that there's a clear distinction between genotype categories. In particular, for every copy of the C allele, I seem to have an increase in both of these phenotypes. But I also see something else that's a little bit subtle and also very important. If I disregard phenotype 1, that is, if I collapse these dots onto the x-axis, I arguably see much less of a separation between genotype categories, indicating that I have less power to discover an association between this genotype and this genetic variation and this phenotype. However, when I expand into the second dimension using phenotype 1, I see a more clear separation, indicating that I might be getting more power from considering multiple phenotypes. So this basic intuition was used last year in at least 3 papers that I know of. The first is a paper coming from Magnus Norberg's group. And in this paper, they show how some classical methodology dating back to the 1960s can be translated over to our current GWAS context and used to perform association. Using traits from the Northern Finland birth cohort, they show that you can actually get an increase in power when you map pairs of traits versus mapping single traits. The second paper is not concerned with doing association, but rather they're interested in using these multiple phenotype models and estimating certain key parameters, in particular the genetic correlation. And they show that by estimating this parameter, you can get interesting insights into the architecture of metabolic syndrome traits. And the third paper is just a computational one coming from Peter Vischer's group, where they extend their GCTA software to be able to handle multiple phenotype models. So this is good. It means that our intuition is probably right. You can get extra power maybe from using multiple phenotype models, and you can get insights into complex traits. There's probably some software available that will do this for us. But there's a major limitation, and that is these classically based approaches are extremely slow. And they're so slow that it really dramatically limits the scale at which we can practically apply them. So let me try to give you some numbers to make this more real. Let's say that I have a cohort of 10,000 individuals and I've collected 1 pair of phenotypes. If I use the GCTA method, it could take up to 4 hours of computational time just to find the model parameters. So this isn't to do the association analysis. This is the first step, the very first thing I have to do to find the parameters in the model. So this might not sound so bad, but remember we're talking about multiple phenotypes, and especially moving forward in the future, we're more likely to have more than just a pair. So let's say that, for example, we have 10 phenotypes. They're all correlated, and we're interested in analyzing each pair of them. Well, now if we do 10 choose 2, which is about 45, each one takes 4 hours, this comes out to be about 7.5 days. So even on a cluster, this is starting to become pretty annoying. And you can see how this is not going to scale when we move up to something like gene expression data, where we have 20,000 phenotypes. So what we do in this paper is to introduce a method for performing this type of analysis, except we make it fast. And what I mean by that is if the standard approach takes about 4 hours, the approach that we propose, called the matrix-variate linear mixed model, or mvLMM, takes about 20 minutes plus 20 seconds. And I'll show you why I split it like that. If I have 10 phenotypes and I analyze each pair, I have to pay an upfront cost of about 20 minutes, and then I have to pay a cost of about 20 seconds for each pair, meaning that my total analysis time goes down to about 35 minutes. So we're going from 7.5 days to 35 minutes, which is a very dramatic speed up. So I want to explain to you how we can achieve this sort of speed up, but in order to do that, I need to give you an intuition about multiple phenotype models, because I think they're not very widely known at this point. And to do that, I'm first going to remind you about how association works in linear mixed models. So here's the basic idea. We collect a trait for each individual, and we put all our traits into a vector. So we have an n by 1 vector, a phenotype vector. We let that be equal to a SNP effect. X is the SNP, beta is the effect of that SNP, and 2 random variables. Epsilon is going to account for our residual error, and it is going to have just a basic Gaussian noise structure. And then u, our random variable u, is going to account for the genetic relatedness between individuals. So the intuition here is that if these 2 individuals are very closely related, let's say they're brother and sister, then we expect that maybe their values of u will be very similar to one another. If these 2 individuals are very distantly related, let's say 5th or 6th cousins, we expect that their values for u are going to be closer to independent of one another. And we can represent that by saying that u follows a multivariate normal distribution with mean centered at 0, and with the covariance matrix looks like this. A scalar, our genetic variance, times a matrix k, our kinship matrix. This is a matrix that encodes all of the genetic relationships between individuals. Okay, so now using the intuition behind this model, that is, using the intuition that we need to account for relatedness between our samples, we want to move forward and propose a multiple phenotype model. And the way we'll do that is, first we'll just take our vector and split it in 2. So now we have 2 phenotypes. The first column here is y1, phenotype 1. Second column is phenotype 2, y2. And now we have an n by 2 matrix of phenotypes. So the first thing we realize from our linear mixed model is that there's the potential for correlation between the rows of this matrix due to the genetic relatedness between individuals. But since we're dealing with multiple phenotypes, we also know there's the potential for correlation between the columns of this matrix. Because phenotype 1 might be correlated with phenotype 2, partially due to genetics, because some of the same genetic factors that affect phenotype 1 will also affect phenotype 2, but also because of environment, because we can assume that some of the same environmental effects that, factors that affect phenotype 1 will also affect phenotype 2. So we're going to introduce a new random variable called E that accounts for that. So what you get from this picture is that we now have a matrix of phenotypes, an n×2 matrix, and I have correlation on multiple levels, first on the rows and second on the columns. And all the correlation between my samples is due to both genetics and environment. So using this intuition, I can build up a simple model. I'll say that y1, our vector y1 and y2, follow a bivariate normal distribution where we have a SNP effect x beta 1, the effect in phenotype 1, x beta 2, the SNP effect in phenotype 2, and then a covariance matrix that looks like this, where this entire covariance matrix is a function of both the genetics and the environment. So if I parameterize this correctly, then I should have the statistical tools available to me to estimate beta 1, estimate beta 2, and then ask the question, are these equal to 0? Which is exactly how you perform association analysis using multiple trait models. And the problem I claim with this is that when we perform maximum likelihood inference using a model like this, it's extremely slow. So if I look at the time complexity, it basically comes out to be something like this. N, where n is the number of individuals, times p, the number of traits, cubed, times t, where t is the number of iterations I need to converge for my maximum likelihood algorithm. So if we consider that p is very small, in this case 2, this is going to be dominated by this n cubed term when n is something like 5 or 10,000, for example. So we wanted to try to improve on this. And what we did was we realized that because of the particular structure of these multiple phenotype models, it's possible to exploit this structure in order to define a matrix M such that I can transform my data and obtain the following distribution, where I have some mean and I have a diagonalized covariance matrix, where each of these D1 and D2 in turn are diagonal matrices. So what this means is, if I do this transformation, I can compute the likelihood of the transformed data in time that's basically linear in the size of the total data set. So if I do a breakdown in time, what I see is that first I have to obtain M, and that's going to make, I have to pay n cubed plus p cubed to do that. Second, I have to compute this transformation given M, and that's going to take me something that looks like this, n squared p plus np squared. And finally, given the transformed data set, I can compute the likelihood, which is only going to take me order np. So if I wrap this inside of a maximum likelihood search algorithm, I can achieve the following time. So I pay an upfront cost of n cubed and n squared, and then for every iteration of my algorithm, I basically have to pay something that's at worst order n, or order n times p squared, when we're considering that p is very small. So the difference between this and this is basically this n cubed term has been brought out, and you no longer have to do this at every iteration. So getting away from the theory and sort of showing you what this means practically, if we apply this to pairs of traits in the Northern Finland birth cohort, this is a cohort for which we have about a little over 5,000 individuals, we have 6 pairs of traits, and we compare our results with GCTA and with respect to time, we find that GCTA takes anywhere from 30 to 40 minutes, whereas the mvLMM method takes about 11 to 12 seconds, conditional on the fact that we had to compute a one-time shared cost of 2.5 minutes. But as you can see, even if I included this cost in each one of these, it would still be a major speed up. So we also wanted to show that in order to achieve this speed up, we didn't have to sacrifice any accuracy with respect to association analysis. So we applied this method to evaluate the association between pairs of traits and all SNPs across the genome in the same cohort, and compared with a method called MTMM out of Magnus Norberg's group. And I'm only showing one QQ plot here for one pair of traits, but this is also shared with all the others. And what you can see is that these results are exactly concordant. In fact, the p-values are have a correlation of 0.99. And we also confirm, as MTMM does, that when you use these multiple phenotype models, you can find about 99% of the significant SNPs that you identified in the marginal model, also in the multiple phenotype model, and you identify new associations on top of that. Okay. So in conclusion, we introduced this method called the matrix-variate linear mixed model, or mvLMM. It drastically, or dramatically, whatever, speeds up the multiple phenotype association and maximum likelihood in these multiple phenotype models. And what I didn't have time to show, but we argue in the paper, is that the ability to estimate genetic correlation and other key parameters in these multiple phenotype models for large cohorts, and the ability to do this very quickly, will be very important moving forward into the future, especially when we consider gene expression data. Oh, before I move on, I also want to mention Last year I published a paper proposing a method for association mapping with longitudinal data. And I just want to say that you could view this multiple phenotype method as both a generalization and an improvement on the previous approach, as longitudinal phenotypes are really just a specific instance of multiple phenotypes. Okay, so I'll move along to my second project. Meta-analysis for model organisms. And the graphical intuition here is that, let's say that we have two separate populations. We could easily go off and perform association in each one separately. Or we could try to combine information across populations in order to perform association. But when we're dealing with model organisms, this kind of procedure might be rather difficult. And so this is sort of what we addressed in this project. So this method was motivated by a particular problem in mouse genetics. So in order for me to motivate it properly, I need to give a little bit of background. So I'm going to start with the idea of QTL mapping in the mouse. So quantitative trait locus mapping has a very similar goal to association analysis. It is to identify regions of the DNA that are related to my traits. And one way that we can do this in the mouse is that we can apply linkage analysis to an F2 intercross. And an F2 intercross is generated using the following breeding scheme. We have two inbred parents, A and B. I represent them being inbred by the fact their chromosomes are the same color. And I cross them to obtain a panel of F1s. Each F1 is going to have one blue, one red chromosome. I cross the F1s to obtain a panel of F2s, and then the F2s have this feature that their genomes are mosaics of the original two founder strains, such that the blocks that they share with the original founders are typically very large. So if I apply linkage analysis to this cohort, I get something similar to an association plot. I have the genome on the x-axis, a measure of significance on the y-axis, and this gives me candidates for regions that are affecting my trait. So this is a very old technology. It's been used very extensively, and I wouldn't want to say anything bad about it. But if I want to be extremely simple, I can boil this technique basically down to having one advantage and one primary disadvantage for my purposes. The advantage is that when we use linkage analysis applied to F2s, we have very high power to detect regions underlying our traits. But the disadvantage is because of the large block structure of the F2s, we typically find very large regions, and they could encompass many hundreds of genes. We call this the problem of low resolution. It means it's difficult for us to find the true causal gene. So there's been a number of strategies that have been proposed that aim to combat this problem. I won't talk about the first two, but rather I'd like to highlight one called the HMDP, which is a panel of mice and an associated mapping framework that was created here at UCLA as a collaboration between Jake Lusis' lab and Eleazar Eskin's lab. So here's the one-slide snapshot of the HMDP. The main idea is to combine classical inbred strains of mice with recombinant inbred strains. Recombinant inbreds are essentially inbred versions of F2s. Then apply a genome-wide association methodology as opposed to linkage analysis. And the result should be that the classical inbred strains provide us with a lot of resolution, and the recombinant inbred strains provide us with high power. And this result is very nicely shown in the figure here, which I took directly from the paper, showing us the association of HDL cholesterol on mouse chromosome 1. So what you see is, you see the linkage peak from the F2s contrasted with the Manhattan plot that you would obtain using, or that you obtain using the HMDP. And what you can see, of course, is that this linkage peak is very broad. It happens to encompass hundreds of genes. And this Manhattan plot has a very distinct peak that is right over a gene that is known to be important for this particular trait. So this picture very nicely shows both increased power and increased resolution. So that's great, and this is a really powerful framework, but there's a few limitations and problems moving forward. And the one that I want to point out is that there still remains very low power to detect regions that have a small phenotypic effect. And furthermore, the ability to increase power is limited by the availability of genetic diversity, or rather the availability of inbred strains. And of course, we can't just create inbred strains because they take a long time, and it's not feasible to do so. So one of the ideas that we had that motivated this project was we said, well, since we can create an arbitrarily large number of F2s, and since, in fact, there's many F2 populations that are sitting in databases, and they have been for years, maybe we can take the HMDP data, combine it with the F2 data, and increase our power that way. And this seemed like a simple enough thing to do, but when we started thinking about the details, we realized that there would be a lot of problems. And the most obvious one is this concept of phenotypic heterogeneity. So if you think about it, we have HMDP data that's rolling off the press right now. We have F2 data that's been in a database for 5 years. it's pretty clear there's going to be a large batch effect between these two data sets, and it's going to make it very difficult for us to just combine the data on the raw level. But there's a couple other things that are a little bit more subtle. One is that there's going to be major differences in the minor allele frequencies between the two populations. So you can imagine that because of the breeding scheme, there's a particular genetic loci which has a low minor allele frequency in the HMDP, whereas it has a more moderate minor allele frequency in the F2s. And so then it's unclear how we can actually combine these two data sets because we're going to have such a major discrepancy. And the third reason I'd like to form kind of as a question. If we have 100 genetically unique HMDP mice, but we have 5 replicates of each, so that we have 500 mice in total, does that have the same amount of information content as if we have 500 genetically unique F2 mice? And if so, or if not, then how do we combine these two populations so that we can take into account this difference in the information content? So in this paper, what we do is to first propose this idea of putting these two panels together to perform association mapping. And then we show that by developing a meta-analysis technique that accounts for the structure in these populations, we can sidestep these previous issues that I showed you on the previous slide. And also we show within our framework that we're able to achieve a theoretically optimal power. And this was published in Genetics just last year. So I don't have time to go into all the derivation. So I've tried to condense the math down onto one slide, but I think it's pretty easy to understand. So we're going to use I to index our population, and this yi is basically going to be our phenotype vector in population I. So this could be HMDP or F2. And I'm going to let yi follow the standard linear mixed model association framework that we typically use. So it's equal to a mean effect, a SNP effect, xi is the SNP in population I, beta I is the effect of that SNP in population I, and then a population structure effect and some error. These follow the standard linear mixed model assumptions we use for association. And then given this, I can get an estimate of the effect of the SNP in population I, which I call beta I hat. And I say that that follows a normal distribution with mean centered at the true beta, which is shared between populations. And its variance looks like qi, which you can see is a function of the kinship matrix, so it's a function of the population structure. So if I want to test the hypothesis that beta I hat is equal to 0, I can form a z statistic, which is just equal to this. And then if I have one of these for each population, I can then use a framework called the weighted sum of z-scores, which is basically I just weight my z-scores by some weights, sum them up, and then normalize by the weights to obtain a meta-statistic. And what we show in the paper is that by setting the weight for each population equal to 1 over the standard deviation of this estimator, we can achieve optimal power within this weighted sum of z-squares framework. So if you know meta-analysis, what you'll realize is this is actually equivalent, this weighting scheme is equivalent to the standard inverse variance weighting scheme used in linear regression-based meta-analysis. The difference here is that we're showing, first of all, that we can obtain optimal power in this framework when we use it, and second of all, that it also holds for linear mixed models when we can optimize these parameters. So we use this method, and we show first that we can get an increase in power when combining these two populations, and we do this through simulation. So what I'm showing on the x-axis is the SNP effect. On the y-axis is power. We expect that as the SNP effect goes up, power is going to go up. You see the red and the green curve are for the HMDP and F2, which we see for this simulation have roughly similar power. When we combine the two through meta-analysis, we get what we expect, a nice increase in power. We also show that by combining these populations, we can get an increase in resolution, and we do this through simulation. So the way we do this is to simulate a phenotype where we know the location of the causal SNP, do a genome-wide scan, find the most significant SNP, and then calculate the difference between those two. And then we basically do this 10,000 or so times and plot the histogram of these distances. So the best method we would expect to be skewed majorly towards the left, indicating that the distances from the causal SNP and the peak SNP are typically very small. So what we see from this is first what we expect, that the F2s have much worse resolution than the HMDP, and then by combining these two, we actually get an increase in resolution for the meta-analysis. We then applied this to a couple different phenotypes. First, to HDL cholesterol, which I've omitted for the sake of time, and second, to bone mineral density. So what you're seeing here is in the first panel is the association plot that we obtained from the F2s. This is going to be similar to the linkage plot. And in the second panel, you see the association plot from the HMDP, and then we combine the two, we get the association plot for meta-analysis. If we zoom in, we see that there are two previously implicated loci, one on chromosome 7 and one on chromosome 4. The chromosome 7 result actually has an increase in significance in the meta-analysis, and it also has a decrease in the associated interval from 11 megabases down to 10. So this result shows both an increase in power and resolution. For the chromosome 4 example, we have roughly the same level of significance for the meta-analysis in the F2s. However, we have a dramatically reduced association interval from 80 megabases down to 8. So this is a nice result to show that we get an increase in resolution. Okay, so in conclusion, we introduced this method for performing meta-analysis with population structure to solve a particular problem in mouse genetics. We think that this can be really very beneficial for people that are doing this type of research using inbred or just model organisms. And it also happens to solve a problem that's similar in human genetics. So you can imagine if you have multiple human populations, each of them has population structure, and you wish to combine them through meta-analysis. As far as I know, there hasn't been a linear mixed model-based approach for doing that. And again, this was published last year in Genetics. Okay, so now I'll move on to my last project. Seems I'm okay on time. And the idea here is that I'm going to introduce a method for calculating gene co-expression in such a way that's robust to a particular type of statistical confounding. So the graphical intuition here is that now we're interested in the whole network, where the whole network is a gene network. In particular, we're interested in calculating the correlation between pairs of nodes in this network, where every node is a gene. So what is the idea of co-expression? Okay, well, co-expressions are used to infer functional relationships between groups of genes. And one way you could view a co-expression analysis is in the following. We perform a number of microarray experiments. Each one is measuring, say, 20,000 or so genes. We calculate the correlation between each pair of genes with the idea that if two genes are highly correlated, they're more likely to be co-expressed, or more likely to be expressed together. If they're not so correlated, then we don't expect that they're very co-expressed. Using these correlations, we cluster genes, and then using these clusters, we attempt to extract groups of genes which we believe to be functionally related. So there's one commonly, or most of the time, I guess, overlooked problem with this analysis that comes in the form of something that people have called expression heterogeneity. And the basic idea is this. If we have a number of microarrays, say n, and there's some non-biological factors which we typically don't know about, and they're affecting the expression across these arrays in such a way to cause the arrays to be more correlated than we would expect by chance, then when we don't deal with these factors in our statistical analyses, this is going to cause major problems. So the easiest one to understand that everybody is likely familiar with is the idea of batch effect. If we have a set of microarrays, half of them were performed in one batch, half of them in the other, and for whatever reason, the within-batch correlation is higher than the between-batch correlation, then this could be a major problem if we're trying to do something like differential expression analysis, for example. The way we can see why this would be a problem when we're doing co-expression, it can be seen by adjusting our data a little bit. So let's take our arrays and let's linearize them, and let's put them onto the columns of this matrix. So then if we introduce some non-biological factors that cause these arrays to be more correlated than we expect, this will be equivalent to introducing correlation between the columns of this matrix. And a simple experiment that you can verify is when you introduce correlations between the columns, you induce correlation between the rows, in effect inducing spurious co-expression. So in this paper, we propose a method for calculating a value of co-expression that's robust to this type of problem, that can correct this problem. And this was published in Bioinformatics as part of the ISMB proceedings for 2011. So again, I'm taking probably a large amount of math and condensing it down to a very small formula, so I'm just going to try to give you the high-level intuition here. We want to calculate essentially a correlation between two gene vectors that's been corrected for expression heterogeneity. So we have a gene vector y1, and we have its mean, so we've centered it by its mean. We have a gene vector y2, and we've centered it by its mean. So RMC could be thought of a corrected correlation coefficient the following way. The top part of this, or the numerator of this equation, is basically a covariance between these two gene vectors, calculated in a transform space using this transformation matrix. And the bottom part of this equation is essentially the product of the standard deviations of these two gene vectors, calculated in a transform space. And the key insight is that the transformation matrix is a function of a matrix K, which we estimate using the observed pairwise correlation between arrays. So remember, the idea is that expression heterogeneity introduces correlation between arrays. So by using the observed correlation between arrays in our transformation procedure, we should be able to sort of correct this effect and get a correlation coefficient that is insensitive to expression heterogeneity. So we compare this approach, which we call the mixed model coexpression, or MMC, with a couple of standard approaches. First is just the basic Pearson's correlation. Second is the Pearson's correlation as applied to a data set, which has been corrected with a method called surrogate variable analysis, or SVA, another method for correcting expression heterogeneity. And so let me show you the first result that I think is kind of interesting. So if we take yeast data and we curate a set of 233 known functional gene modules, so that is groups of genes which are known to be functionally relevant. And then we calculate a statistic for each of these groups based on their pairwise co-expression values and then an associated p-value. If we look at the cumulative distribution plot for these p-values, a perfect method would have a line shooting straight up at 0, indicating that all the p-values are highly significant because, in fact, all these groups of genes are related. We don't see that for any of these methods, but what we do see is for the MMC method, there is an enrichment of p-values very close to 0, or very significant p-values, indicating or suggesting rather that by using the co-expressions coming out of MMC, performing this procedure, you can get an increased power to identify functional gene modules. The last two results, which are in the paper, I didn't put here just for brevity, I want to highlight very quickly. The first is that we show that by using MMC, you can do a better job of prioritizing truly functional gene pairs. So you can imagine that if you calculated all pairwise correlations between genes, what you would like, and then rank ordered them, what you would like is for the most truly functional gene pairs to be highly ranked within this ranking, or highly prioritized within this ranking, rather. And so we basically show that MMC does a better job of highly ranking these truly functional gene pairs with respect to the other methods. And then the second result is pretty simple. If we have two data sets which are exact replicates of one another, just produced at different times and say in different labs, and we compute the correlations in each one and then look at the concordance between them, we show that the MMC method has a tendency to produce more concordant results between these two, indicating that it's less sensitive to to experimental specific conditions, for example. Okay, so I'm not going to give conclusions for that. I'm just going to jump straight to my PhD-wide conclusions. And I wanted to come up with, I don't know, something kind of simple, like, grand to say what I had done during my PhD, but instead I limited myself to 3 sort of practical bullet points, I think. So the first thing is, I think that most of the stuff that I've done can be summarized using the first bullet point. I developed methods to increase power to identify relationships between entities in complex systems. So the obvious one is increasing your power to identify an association between genetic variations and phenotypes, or identifying, doing a better job of calculating a correlation between two things, which is also something in the multiple phenotype paper, which I didn't have time to get into. The second bullet point is more practical, and I feel that my work will, or hopefully has, enabled researchers to use their data in a more realistic framework. And so, as I kind of mentioned previously, that big picture that I showed you that cost hundreds of millions of dollars to create, there's a lot of data that's just sitting in a database somewhere and that's really being underutilized. For example, multiple phenotype data. Most people have only used one phenotype and mapped genetic variations using that, as opposed to considering all the information together. Or longitudinal phenotypes haven't really been exploited, or at least I haven't seen a lot of literature exploiting them. So I hope that the work that I've done will enable researchers to be able to take these data sets, use them to discover more, and then use this in a feedback mechanism to create even better studies and to do a better job of discovering, basically. And the third is more of like a future work thing, I guess. Since I started, I've been thinking about this idea of risk prediction, and I feel that every project I had, I always was sort of thinking about how to do genetic risk prediction using this, extending this, moving this forward. So I hope that some of the work I've done, at least in my head, has laid a foundation for a future work in risk prediction, which is really what I think is a very important aspect of genetics. So with that, I want to give thanks to everybody. I have a lot of people, so there's no way I could fit everybody, but I tried. So I want to thank my co-authors for my papers that I mentioned. I specifically want to thank Hyun Min Kang. So Hyun was a graduate student in Eleazar's lab when I started, and he really took me under his wing and showed me the way of linear mixed models and all sorts of stuff. And so I really appreciate that. I feel like I was a little kid that didn't know what was going on, and Hune really kind of helped me a lot. And then I'd like to thank my funding sources. So I was funded on the Genomic Analysis Training Program, or GATP, for a couple of years, and then just this last year on a neurobehavioral genetics training grant. The GATP in particular was a very influential thing for me because I met a lot of people. I just had a lot of good interactions, took a lot of good classes. And I'd like to thank from this training grant, Ken Lang, Janet Sensheimer, Rita Cantor. These were people that were that I think really helped me a lot during my time. And then I'd like to thank my committee, of course, Jake Lusis, David Heckerman, Chris Lee, Amit Sahai, and my advisor, Eleazar. I have to single out Jake because, our labs really collaborate a lot, so Jake has been this kind of constant figure during my PhD, and I just want to say that you're definitely one of those people who has been and will remain to be an inspiration for me, moving forward, and I really appreciate everything you've done for me. Also want to thank David. I didn't know David, I guess, really that well until 2 years ago. He was this sort of mystical figure that would, be around every once in a while, and I was like, I've heard of David Heckerman. And a couple of years ago, I had the pleasure he was on, agreed to be on my committee, and then I had the pleasure of working with David at Microsoft. And since then, I've gotten to know him, and I think, David, you're a great guy, and I really appreciate, the opportunity to work with you, and I think you've really taught me a lot, and I appreciate that. Also, I want to thank Chris Lee. Chris and I haven't really worked together, I guess, but I took his class in bioinformatics, which is hands down one of the best classes that I had at UCLA, it was very difficult. And then later, I did, yeah, it was very difficult in a good way. And then later I found that when we talked, we actually had a lot of the same opinions about things and have a lot in common. And I really, really appreciate you being on my committee as well. I'm going to save Eleazar till later. So my friends at UCLA, so I want to thank Emra, Aicha, Mete, Farhad, Joseph, and Luz. All these people were really great friends to me and also really great colleagues. We talk about personal stuff, and we also talk about research. And I'm not going to embarrass her, but obviously Luz is very important to me, so I just gave her a bold and a star to indicate that. And so I also want to thank my family members, which most of you don't know. My grandparents, Peck and Gracie Daniel, hands down two of the most important people in my entire life. My mom, Nina, my little brother, Austin Furlotte, my Uncle Kenny, Aunt Lisa, Uncle Al, Aunt Carol, and then four of my really good old friends, Yuki, Ples, Peter, Doug. Hopefully if they see this on YouTube, they'll know that I appreciate them very much and that I wish they could be here. And then I saved Eleazar for last. I don't want to get sentimental or anything. I don't want to go crazy, but I just want to say that, these last 5 years have definitely been interesting. I think I've changed dramatically from both personally and professionally in the time since I started. And I feel that I've become a better scientist, a better critical thinker, a better writer, presenter, all these things. And I really appreciate the time, the effort, and all the patience that you've had with me and just everything you've done for me since I started here. So thank you. Thanks.